in this talk
This meeting is being recorded
from different parts of the world. So I don't know what time it is now, but we are having a very pleasant evening here. I'm pretty sure that we all are really tired of this pandemic. It's taken a big toll on us. While I'll be talking about the latest developments with regards to the pandemic today, I really hope, pray, and wish that we get rid of this pandemic so that we can get back to our balanced, happy, normal lives. So with that, I would first like to thank Dr. Sudha and Dr. Raja for giving me this opportunity to present on this platform today. So in this talk, I'll be talking about genome surveillance, why genome surveillance has been important throughout the pandemic and why it will continue to be so, not just with regards to SARS CoV-2, but also with the other viral diseases or any other infectious diseases that we might have. So just to provide a deeper understanding of why genome surveillance is essential for infectious disease, we need to understand that there's a kind of communication that happens. So you've seen that throughout the world, there are several organizations that have been involved throughout the pandemic, be it the World Health Organization, or in India, the National Center for Disease Control and the Ministry of Health and Family Welfare. So the main reason why we need genome surveillance is to keep a track on what kind of variants are coming up, whether they are more infectious, whether they're spreading more fast. And this is mainly important because it provides public awareness. So with the kind of genome surveillance that is available, public can be made aware of what steps are to be taken. It also prepares the medical people to prepare infrastructure. It also helps build resources. And with regards to the governments worldwide, it helps them take decisions. The most important thing about genome surveillance is that it helps us understand the kind of variations that come up, which would be important and decide the kind of vaccine development that we have. And this we have seen throughout the pandemic, how it has helped us get going, do very fast, take fast decisions and come up with vaccines which have been rolled out. And now people have been vaccinated and they've developed herd immunity. So it's also important because the kind of, as long as we keep sequencing various genomes, particularly now in this case, SARS-CoV, several genomes have been sequenced. And I've just put up an example here of one particular study where they found that a variant showed up in a community transmission case where one of the variants showed a mutation in the ORF7A, which had a kind of growth defect. So this kind of a finding helps us understand the virus better. And it also provides us a way to go forward where this particular phenotype can be taken into account when we develop vaccines. So as far as the population throughout the world is concerned, there have been several instances. I mean, we know what has happened throughout this pandemic. Too many infections, a lot of fatalities, which was really unfortunate. And initially, the genomes were getting sequenced, but not at the rate that it had to happen. But after a certain period of time, it did start picking pace. And this picture I've just put forth, it's not a very updated one, but this is what I found at NSACOG, which is the Indian SARS-CoV Genome Consortium. We have been able to sequence a lot of number of genomes for the SARS-CoV-2 virus. And there is a representation of most of the variants that were reported throughout the world. So while this is important and we need to do more and more sequencing, I'm pretty sure a lot is being done throughout the world. I would like to touch upon now what sets us apart. Why does we being a genomics service company, why are we better equipped or better armored to provide any inputs or any help that we could with regards to genome surveillance? So the kind of sequencing platform we're using for SARS-CoV is the nanopore. And the best thing about it is that it has the fastest turnaround time, which is approximately about 24 to at the most 36 hours. And I can vouch for that because we have been able to deliver results even less than 24 hours. So starting from even the nucleic acid extractions to the deliverables where we provide you with the lineage data and the variant calling, it just takes approximately that time which I mentioned. What has been in a very standardized way, there is a pipeline in place for the SARS CoV-2 genome sequencing at Genotypic, wherein we use a protocol which is already published. It's called the Midnight Protocol. So what it does is we have a panel of primer pools which span the entire genome sequence of the SARS CoV-2 virus, and it provides us amplification for about 1,200 base pairs of each of the genomic region. So since it's a tiling array, there are pools which provides amplicon that have a little bit of overlap with each other. So once all the PCR is done, the entire genome is sequenced. So you have a representation of each of the region from the virus genome. The main reason it's called midnight is because of the size of the amplicon that the primers provide. It's 1200. So once we have that panel designed, whenever there's a clinical sample available, the RNA extraction is done. We do the cDNA synthesis, and then it's taken up for a multiplex PCR, wherein the pool 1 and pool 2 primers are used. So once this PCR is done, there is library preparation wherein the barcoding is done, and also the adapters are provided. So once all that processing is done, it's loaded onto the nanopore machine where the sequencing happens. And the advantage of this is that there's also live base calling. So while sequencing itself, the bases are called. Once the sequence is complete and there's enough data, it's taken over by the analysis team in the bioinformatics division. And that is where the entire analysis workflow is carried out. Now, let me come to another awesome part of what we are equipped with. So this is the brainchild of our CEO, Dr. Raja, and he has come up with this software called Commander. It's an integration of many tools, several domains that coordinate and come together to provide an easy analysis solution. So we have used this tremendously for the SARS CoV-2 sequencing analysis. And when the data is ready and it is provided in the FASTQ format after demultiplexing, and it just takes about 20 to 30 minutes to provide results, which include the lineage data, the variant calling, and if needed, even the assembly. So this is just a picture of what the workflow looks like. So when the nanopore sequencing is done, we get the raw reads, wherein there's a tool called pore chop, which actually chops off the adapters, and then you get the processed reads. These processed reads are mapped to the reference genome. Here in this case, it's the SARS-2 COVID genome. And once we get the BAM files, which are the alignment files, they're provided and fed into the SAM tools, which help with the variant prediction. So this workflow has been standardized and it runs smoothly. And as far as the lineage prediction is concerned, Commander has integrated at least three different online tools which are available for lineage prediction: the Pangolin, Clade, and the Scorpio. So what you see here on this slide is an output of the lineage prediction results, wherein we see that, like before, I have told you that we have sequenced several genomes, and this is a representation of what it looked like in the last 2 months. So we mostly got the Omicrons here. So while we do get the results for SARS-CoV, it's also important to tell you that we have optimized this kind of a pipeline for for other infectious disease as well. Most of these are viral diseases. So apart from SARS-CoV, we have panels designed for the human adenovirus, the rabies, which is still persisting because people fail to get their dogs vaccinated, and norovirus, which is not very common, but it does show up. I'll be touching up a bit more on the other two diseases mentioned here, the Cassino-Forest disease and measles. So measles is something which has been dealt with time on and on. The most recently we found that in 2018 and 2019, there was this outbreak in the US, which put the entire world in a state of confusion or chaos. It was mainly because it was due to the outbreak in a small community where the kids were not vaccinated. So they were susceptible. And it started with their visit to a recreational park, and it spread from there. So it put the entire population, it made it very susceptible to getting this disease. So it was predicted towards the end of this outbreak, it was predicted that there would be 1 in 4 among patients that will need hospitalization. So the only solution for that would have been a 95% vaccination to get herd immunity. So in order to achieve this, we really need to understand that if a virus is able to replicate freely in any host, it will lead to different variations, which probably could get selected and then cause a major pandemic. So this has to be kept in mind. So I would like to emphasize here that we do have a panel designed even for measles. The other disease that I would want to reflect upon is the Cassino-Forest disease. It's one of the neglected diseases. It's also called the monkey fever. It's spread by ticks, which are infected, and they transmit the disease to virus. So it's an arbovirus, flavivirus family disease. The main reason this has been neglected that it has been sporadic. So many people might be getting it, but there is no awareness. But we do have clinical samples coming in for this virus. And most recently, we were able to analyze some samples. And in that, we found that there were several mutations in the polyprotein gene, which suggests that the KSD virus is undergoing change. It's mutating. So if this is not kept in mind and there's not a surveillance mechanism in place, we could have a potential huge pandemic or an outbreak here. And we do have a panel again for this disease as well. So while all this is there, what is important to know that there is always a devil in the genomic detail. So in order to stress upon that fact, I would like to just tell you a few things about how a mutant is selected. So in nature, for any infectious disease or a virus, it is survival of the fittest. Whenever there's a variation or a mutation, it is either a positive impact or a negative impact as far as the disease agent is concerned. So in any case, a virus would never does not intend to kill the host, because it will be against its ability to transmit further, because it really wants to grow in the host so that it can transmit or jump from one host to the other. So the positive impact would be like a more transmissible variation. The other negative impact that might come in if there are other variations is there's an increased severity in the infection, wherein the hospitalization rate or the death rate among, if it is an animal virus or zoonotic virus, then there's a suppression of the virus at that stage, wherein if the host is killed, there's a lesser chance for the variant to get passed on to the next host. So with that, we have seen the appearance in this case for SARS CoV, several variants, some of which were talked about while some were not. It was mainly because there were some which really showed a positive impact on the virus, increasing its transmission rate or the severity of the infection, while there were others which were not put in the list of concern, mainly because they had no impact, and some were in fact negative, where they killed the growth of the virus. So this is just, I've just put up a list of what I found from the Center for Disease Control USA, where they have categorized the variants for SARS CoV-2 in different ways, where one type was the variant being monitored, which are still on because they're still circulating in the population, not at a high level, but there's still these virus variants are still in the population. Then there are variants of interest and variants of concern, in which we see the Delta and the Omicron, because Delta did cause some increased severity in the disease. Omicron, as we all know, has been more transmissible and it spread very quickly. It was first reported in October 2021, and by January, February, the entire world was in the grip of this particular variant. There are yet no variant of high consequence, which would mean that these viruses actually are really infective and probably are very fatal once you get norovirus disease. So with that kind of an understanding, I would want to touch upon what is important for the SARS virus as far as its infectivity is concerned. So I hope most of you know that in all the variants that were of concern, the spike protein was what was talked about. So if you look at the alpha, beta, here I've put the spike protein for the variants that were listed as variants of concern, the alpha, beta, delta, and omicron. Here the spike protein showed changes, mutations, and that led to the changed behavior of the SARS COVID virus. So just to tell you that the spike protein is important because it helps the virus interact. So there is this ACE2 receptor, which the spike2 protein interacts or binds to, and it helps the virus penetrate the host cell and cause an infection. So that lets us to know what kind of an genomic epidemiology we should expect from this kind of variation. So how do genomic variations in any infectious agent affect the disease? Mainly, what happens is either the transmission or the severity or the pathogenicity, like how well the virus is able to, how quickly the virus is able to enter the respiratory system and take over the entire system and cause a lot of severity in the infection. So in that respect, because of the genomic surveillance or genome sequencing, it was found that there was this particular mutation in the spike protein in the B117, where the N501 amino acid was changed. And this led to a mutation in the spike protein, wherein it was able to bind to two different molecules or two ACE2 receptors. That helped the virus penetrate quickly and cause more infection. Now, I'm pretty sure you're all waiting to know more about what happens with the latest variants that have come up in the news. So if you recall, just a few, maybe a couple months ago, we did hear about this Delta Omicron recombinant, wherein there was talk that the Delta and Omicron, just to understand, any person could be co-infected with different strains of the virus at a given time. This has been reported, but not all of these co-infections have come into light. Wherever there was a Delta and an Omicron infection, there was there were some incidences where it was thought that it had recombined in the host, and then it had led to the appearance of a new variant, which was nomenclatured as the Deltacron. But there was no evidence per se to say that this particular variant was more infectious, more transmissible, because as early as it came, there was it was not in circulation, and it was not talked about a whole lot. There was just some concern that this has happened. But later on, there were also debates to suggest that it was mainly because there was some sequencing artifact. So I'm not saying that it it is that, but there is a remote possibility that during the sequencing reaction, some reads get assembled in such a way that you get a recombinant type, and that comes up as an artifact suggesting that it's a new variant. But as far as the Omicron, the recombinant version of the 2 Omicron strains, which is being categorized as the XC variant, is concerned, there is some possibility that there might be a true variant. This I speak with some exercise that I've been able to do. So what is being said about that, about the Omicron XE variant, is that the parental strains BA1 and BA2, when a person had a co-infection with this, they were able to recombine in certain regions of the genome, and out came the XE variant. So that exercise with the team has helped us put up a small result here, wherein I'm displaying the XE spike protein, wherein if you look at the strains displayed here, they are the 2 parental strains, the BA1 and the BA2, along with the new variant, which is XE2. The circle regions show that the XE2, though it's varying, does show a commonality with one of the parents. So if there is a region of change, it is either matching the BA1 parent or the BA2 parent. So as far as the XE spike protein is concerned, there is no change, and hopefully that should help us feel at peace as far as this is concerned. So in order to understand it more, I did try and put up a recombinant map for this particular variant, wherein the genome, so the green bar represents the BA1 parental genome, and the red bar is the BA2 parental genome. So based on the sequence changes that we found in the XE sequences that were obtained from the different the data that is available worldwide. So at this stage, I would just like to tell you that whatever sequences are available for this XE strain are mainly from the UK region, and they come from only 3 labs. So there's not enough data out there to actually go into the depth that would be needed to really understand this XE variant. So if you look at this map here, there is some kind of recombination that we see here. The red region in the middle bar is where the variant has probably recombined with the BA2 parent. And these are also the other regions which represent the BA2 parent in the XE. Whereas the green ones are coming from the BA1 parent. There are 3 mutations based on what the data says. There are 3 mutations which are unique to the XE variant. And these fall in the ORF1A, which is the protease, and one of them is in the ORFB, which is the replicase. It probably falls in the polymerase part of the genome. So while these changes have occurred and there is recombination based on what we are seeing here, based on the sequence data, what this translates into as far as its behavior is concerned is something that we are not aware of yet. It's being said that the XE variant has signatures of both the parental strains. There is, and as I have shown you in the previous slide, there is a unique feature in some of its genomic regions, but it's like only 3 of them right now. There is also data to suggest that it's 10% more transmissible compared to the BA2 variant. Had this been the case, we should, if I'm correct, this particular variant was first reported in the UK in around January, I think early January. Had it been more transmissible, we should have been able to detect more number of cases as far as the XE variant is concerned. But that has not been the case, and we don't see it as much. I did show you that the spike protein, although it's a recombinant version of the BA1 and BA2, it does not show any structural changes in the protein. So there is no data to suggest that this might actually lead to increased transmission or severity of the infection. Some reports do suggest that this particular variant has a slight growth advantage over the other variants, but there's no concrete data to support this view. So while we are debating its existence, can we just reflect upon what could be the XE? Could it be an artifact? Well, I wouldn't negate the possibility, mainly because whatever data is available for the XE variant has come from the short read platforms, wherein it was either the Illumina and some data coming from Sanger sequencing. So these are short reads, wherein it depends on the number of reads that are represented from a particular variant. So suppose if a person was co-infected with the BA1 and BA2 variant, and for some reason the viral load of one of the variants was more, it would get represented in the sequence because those particular reads would be then more represented. So when the sequence is assembled, it would actually hamper the kind of assembly that we obtain. So if that is the case, then it could possibly lead to a sequencing artifact, suggesting that this is again a recombinant variant. So we should keep our eyes open to such a possibility and not just take this, the occurrence of this variant as such. With all that I have spoken to you about, I would like to just reflect upon these few questions. Will the XE variant be more transmissible? Well, as far as the reports and the data is available, there's no concrete evidence for that. But since it's a recombinant strain, we'll have to just keep our watch and wait out. Will it cause more severe infections? Mainly the infection-related cases have been associated with changes in the spike protein. And if the words of well-known virologists are to be taken into consideration, they are of the view that if a variant is showing characteristics of the parental strains, then it would not behave any more differently from the parental strains. So let's just hope that this is not this is the case with XE variant, and it does not show more severity as far as the infections are concerned. Will it escape detection by RT-PCR? Well, since it's a recombinant, it's a possible recombinant version, and we are constantly getting feedbacks, inputs on this data, there is always a possibility that the kits can be updated, the primers can be remade. And so if this is the case, we should be able to quickly get around this difficulty and be able to detect the variant. Will it escape the vaccine? Well, that is still in consideration because we do not have many cases with this XE variant. Let's just try and get the data out before making any conclusions. With those points discussed, I'll leave you with the slide to reflect upon. Thank you all.